Thank you so much for joining. So these are all the frequent webinars, which we usually used to have Wednesdays. But we have been long time associated with Reproducibility, which is a wonderful organization. Reproducibility is spelled REPRODUCIBILITEA. TEA is the normal this thing. So they work on a premise that we need to exchange science at the cross borders, and the science should be exchanged in such a way that we need to reproduce the works that we are trying to do. So there are about reproducibility chapters across different levels, different organizations. Largely in Europe, there are about 6 chapters. Bioclues has been there since 2016. Ever since I moved back to India, and we have had subsequent programs with them earlier, but not at this particular level. So today we are going to have Indrani Biswas, who is a joint secretary, and she's an ICMR PhD fellow in Pondicherry University, and then she's going to give her talk today. Okay, there will be a good amount of Q&A session, and please ensure that you interact well. And thank you, Rachel, for recording this particular talk in advance. Please, Indrani. Yeah.
Thank you, sir. Thank you, sir, for such a polite introduction. So I shall share my screen. Is it visible in entire screen?
Yeah, it's visible.
Okay, thank you. So, Prash sir has already given a brief introduction about about myself. So, I would just like to add a few things upon it. So basically, my area of interest is cancer biology and drug repurposing, for which I have been working for more than going to be 10 years. And I have worked on a panel of cancer cells, studying with liver cancer, lung cancer. Now, and my PhD area of research is specifically focused on glioma, which is basically malignant form of brain tumor. There are more than 135 varieties of brain tumor, but specifically I'm working on glioma. For today's talk, I have chosen this particular topic, drug repurposing implications in cancer biology, never-ending tale. To start with, I must say this particular term has been acquainted with to most of the cancer biologists or the cancer scientists more than 150 years, because this concept has been prevailing for such a long term. Despite we see so many news coming up, like the most important is, will cancer get cured? What will be the deadline? So all scientists across the world, across the globe, are fighting across so that we can find one-hand solution for it. So to start with this topic, I have taken this particular concept, that is a process of drug discovery, a conventional route. So if you look back to the basic features of developing or designing a particular drug, what is it actually, what was the basis, and how it has come up to this level. So it starts with a very small group of molecules that are termed as hits. So these hits will have some features, some chemical features, okay, but in order to take that to define it as a particular drug, it has to pass through several stages. And that span about more than 10 years, if you consider this step of developing a proper drug. So it passes through several stages, and these hits these are, as I mentioned, like these are specific chemical moieties or molecules that have some features which helps in interaction with some other chemical moieties also. This interaction can be with the biological macromolecules that we know commonly as a DNA, RNA, or proteins. When this heats combines with these targets, or this say suppose these heats have some effects on these targets, it comes up to the leads. Okay, but to take up that stage from leads to drug, it has to have several features. It has to have several features. The major thing is safety assessment, and the next thing is a dosage optimization. So these are basically the dosage is specific to each type of cell, to each type of organism. It varies. Okay, so that what do you define by a dosage? It is a particular concentration or the amount of this particular compound that you are giving, through which your biological system will give some response. Okay, so this response can have several effects. So one of them is actually severe toxicity. Okay, so to ensure that the your it should be safe enough, it should not make huge amount of toxicity. It has to undergo the safety assessment. So in this particular umbrella term, safety assessment, you will have n number of laboratory tests or pharmaceutical tests. You have systemic toxicity, you have oral toxicity. All this will ensure how much effective or to what extent that particular compound will be effective also, and also it should not produce any kind of adverse effects, or even if it is produced, it should be tolerable when it comes up to the humans or any other organism. So once it crosses all these phases, it achieves a term called drug. But then the journey doesn't end here. What is it? The next factor that determines how it should be effective on the organisms? So here comes the authority, like Food and Drug Administration. So they ensure like this drugs should pass through several stages. So one of them is the basic fundamental will be subclinical, or some people also refer to as preclinical. But I would like to put it in a different way. Why not preclinical and more subclinical? Because it is in the intermediate stage before going to towards the market. It should be applicable to the population. At the same stage, it should bear all the features that should enable, like against which it has been developed, like for a particular disease, like in this case is cancer. So going through these stages, subclinical, phase 1, phase 2, and phase 3, it goes on to the phase 4, that is the drug is launched to the market. So all the patients will be taking that or consuming this drug, and post launching also there will be FDA will make sure post launching there should not be any adverse effects coming up. If the effects goes or increases, then FDA puts ban on those drugs again, and they withdraw those drugs back to the companies. Okay, so what is it subclinical? I'll come up to that. So basically it is a stage before it launches to the group of human beings in phase 1. This has to undergo the subclinical, where several models, experimental models are employed. Okay, and this phase 1, phase 2, and phase 3 will comprise your voluntary group of population who are willing to take the test of running these drugs, so that we can find out how far, to what extent it is effective or efficacious. Okay, so this is a very basic concept of drug repurposing. how the drug repurposing concept is utilized, how it is being taken into action, and what all can be its basic implications or rather applications. So, based on your literature review search, yes, without that, you cannot move a step ahead. You have to do thorough literature search for a particular cancer when you are dealing to, I must say, because for each cancer types, you have several different morphological, histological, molecular, cellular pleomorphism. So pleomorphism means features are variable from one cancer to the other cancer. So one drug may be applicable for one cancer type, it may not be applicable for the other. And then, yes, you have to screen drug libraries. Here, while doing the screening, you have the most powerful tool that we have now is AI and computational biology also. So using all those tools, you can screen basic sets of those drugs which might be effective against that particular cancer. And based on those drugs that might be showing efficacy, you try to um, enhance its effect on a particular disease. It can be a new disease, it can be a rare disease, and it can be one of the neglected disease. So rare disease, I hope people are aware of the term neurodegenerative diseases. So neurodegenerative diseases are now, most of them come under the category called rare diseases. And neglected disease, which has been existing for years, but it has not come into the alarming notion of the scientists. So, and so this concept of drug repurposing, basically it is, repurposing means you are changing, you're trying to change the effect of that particular drug with respect to a particular disease that you have chosen. Its application will change, its mode of action will change, how it is going to work against that particular new disease. So that's why we reposition it, or we redefine the purpose for which the drug has been made. So as we saw that how the traditional route of developing a particular drug, that it takes more than 10 years, 10 to 17 years. So in compared to that and your drug repurposing, there are few features to which this drug repurposing concept has become a hotspot in drug discovery field. So many drugs has been, as I mentioned, like going over to the phase 4, like once when it has passed all those 3 stages and it comes up to the market, FDA decides, like if the adverse effects are increasing in a particular population, they try to withdraw it. So they may be failed or they may be banned. The other thing is resources and the time expenditure. The time that has been taken to develop a particular conventional drug, it will be reduced as in compared to our repurposed drugs, because we already know what all side effects it has, what all effects it can induce against each type of diseases. We all know how it can work, but what is it maybe newly residing here, right? So in that way it can save the resources, the financial burden of the company or the pharmaceutical labs who are trying to design a new repurposed drug. And yes, considering this multi-targeted therapy, so targets, it has been decided like this particular drug, it is specifically binding to a protein, and it is helping in reducing its the propagation of the disease. But it might have a new target in that case. So we can take a group of drugs or one drug choosing against a new disease. What if it can show some efficacy against that new disease with a newly defined target? And obviously, considering the fact it has passed through the major safety profile assessment, it reduces the efforts to redefine its toxicities or the adverse side effects that may be associated. So in such case, you no need to refocus it. We have all this information available with us. All we have to sit is to do a thorough literature search. Now that the tools like AI, machine learning models, or you have molecular docking softwares, you have simulation softwares, you have supercomputers who can effectively screen those compounds. You can get the results within a day or two and can use it for several diseases, say suppose cardiovascular disease, or if you want to define it for neurodegenerative or for diabetes mellitus, or else you can take some painkillers or simple analgesics and can apply for infectious diseases also. So the era of drug repurposing is not restricted to not only cancer biology but in other fields of healthcare. So, coming to the very crucial concept of cancer, we all know how it is. It is all like it spreads all over the body. It origins from a benign tumor, and it undergoes several sets of mutations that uh, drives its control, and it undergoes extensive growth. It bears higher proliferative index. It tries to evade the basic mechanism of a cell death, which is widely coined as apoptosis, and it tries to evade this immunosurveillance mechanism. So the immune surveillance mechanism is all about our immune responses that we have, comprising our T cells. You have the antibodies producing B lymphocytes, right? So the T cells, you have several subcategories. One of them is your helper T cells. The other you have cytotoxic T cells, and then regulatory T cells. These three people plays a major role. Uh, Regulatory T cells, they will try to suppress the antigen presentation of those cancer cells, and T cells, helper T cells and cytotoxic T cells will try to produce the cytokines so that it can activate the macrophages or the antigen presenting cells, and thereby once antigen is presented it can be destroyed by the macrophages or the monocytes and then macrophages, right? So all this is being involved here. So our this malignant tumors or the cancer cells will have all these features that can evade all of these responses, and once it starts to bulge from one particular area, it will try to break the endothelial lining from one organ, and it passes to our fluidic system. It may be lymph, it may be blood, or it may be CNS fluid. So it tries to migrate. Those cells will try to migrate into each of these fluids, and once it starts this process, it is an ever-ending process there. So it will try to migrate through this fluid, and it will try to reach your other organs or throughout our body, because our body is all mainly composed of this tissue fluid, right? So that is the stage where it comes as the migration and the invasion. All that it has started now. Now it tries to go and incorporate into a second organ or a secondary site. So there what happens is the endothelial cells undergoes a reverse transition. They undergoes to mesenchymal to epidermal transition, MET. So while breaking in the primary site, you have EMT, and at the secondary site, you have MET. So all these features, now that it has undergone the MET, it will try to proliferate and grow in that particular region and also spread to the other organs or the nearby regions. Now it has been reaching to your grade 3 as we define, like that is the grade 3 stage. And finally, when you have your entire body, it has been covered by all of the organs, or 90% of your body are covered by the cells, we tell that it is the final stage or the stage 4 it has reached, right? So, if we take a deep look into these tumor types, we can categorize them the other way. They are called solid tumors or liquid tumors. Now, what are these solid and the liquid tumors? So, these solid tumors are the tumors which are originating from your tissues, all kinds of tissues or organs. Maximum cancers that we have is based from the organs that we develop. But this about the liquid tumors, it is mainly from your blood or the lymph that is generated. Our blood, when we have leukemia, it has several complicated markers. Differentiating them on just based on the histopathological basis is extremely difficult, because there are some subsets of these cells which we call as stem cells that play a major role here. When we see, if we take a deeper look into the embryogenesis or the developmental biology of the embryo, you have the stem cells which is responsible for the development of an entire organ. But during these cancers, the stem cells undergoes a series of changes that they develop the cancer stem cells, and this define how these cancers should behave. So, considering the picture, like you have a complicated system of your this mechanism of the cancer development, and at each stages you have distinct sets of genes and those proteins that makes it so challenging. In this respect, there are some cancers. There are some. Hello. Hello. What?
Nothing.
Is it fine?
Nothing. You just continue. You can just continue, please.
Okay. So, considering the picture, how these cancers can be so difficult and be complicated, for past several years, here I have jotted down few of the repurposed drugs that has been taken, that has been considered based on the mode of the action they have exhibited in different other diseases, and later on they have taken to the cancer treatment. For example, you have here cyclophosphamide, which we are all aware of, is basically an alkylating agent. It was firstly developed as an immunomodulatory agent, which suppresses the regulatory T cells. But it was later they took this idea, like we can try it on breast cancer treatment, and its mechanism or the mode of action was like, based when this drug is entering in our system, this alkylating agent is responsible for an SNP. So we have this category of enzymes called cytochrome P450. So there is one SNP which actually promotes the conversion of this cyclophosphamide from its inactive to the active form, and that's why this particular drug will be more effective. So, taking this particular notion, they thought like they can take it for the breast cancer treatment. Likewise, we have tamoxifen, which was basically designed as an estrogen receptor modulator. So, it was used to treat Albright syndrome, which the basic mode of action is it acts in a competitive manner to bind towards a receptor, the estrogen receptor, and in the same way, like I discussed for cyclophosphamide, right? It was able to undergo the conversion only because of the SNP. So, based on this concept, it was being considered for breast cancer treatment. And you can see the other set of drugs which they have been utilized for directly, like an antibiotic doxorubicin, which is a gold standard drug used to treat lung cancer, and it has been also been taken for other cancers as well. Likewise, we have aspirin. It's basically an anti-platelet agent or anti-pyrogenic. So pyrogenic means it acts to reduce your fever. Okay, so although it was being utilized as reducing your fever, it has been implemented in cardiovascular disease to resolve clots, because it's an anti-platelet agent, because platelets helps in blood clotting. So taking this particular notion, they thought, if blood clot is one of the mechanism that has been resolved by aspirin, it might have some other antifibrinolytic effects when it comes to taking for cancer treatment. So they have found, interestingly, it downregulated AMP kinase 1 and mTOR. So it's a basic signaling pathway that you have from PI3K, AKT, and it goes and regulates a transcription factor called mTOR, which is mammalian target of rapamycin. And this particular signaling molecule, you can find it is upregulated in several cancers. But again, the type of mutations that it is present in each of the cancer types, it is wide and very difficult to identify. So here we have again thalidomide, which is basically a cytostatic and again immunomodulatory agent. So cytostatic, you have two category of drugs when it is coming to kill any cells. So one is cytostatic, that is, it is static means it is in stagnant or stationary phase, and cyto means cell, and the other is cytotoxic. So if it is cytotoxic, it is like inhibiting the proliferation of that particular, proliferation means increase in the cell number. So those agents will act in cytotoxic. And the other way is cytostatic, means it is inhibiting the growth of the cancer cell. So firstly, it was utilized to treat leprosy, which is also termed as Hansen disease. But then later on, it has been implemented to treat multiple myeloma or myelodysplastic syndrome, which are basically adverse forms of your blood cancer. Okay. Likewise, we have anthracyclines. So it's a class of antibiotics again. It is cardiovascular disease, basically acted to resolve CVDs. But then again, it has been seen to resolve a type of non-malignant tumor called hemangioma. It is present in kids or children who, after within a year, they have some clumps of blood vessels on their arms. Okay, so it was being seen to reduce this particular. It was shown to be effective against this particular disease, and it helped in reducing cardiotoxicity and also improving the left ventricular function with respect to the CVD modulator, which accompanied as a chemotherapy drug. That means it was given in the form of chemotherapy to the cancer patients. It was seen like it didn't induce any kind of toxicity with respect to the cardiac function. Okay, then we have like valproic acid or phenothiazine. These are called as antipsychotic. So basically, this kind of drugs, they have come up newly in the market. Antipsychotic drugs works basically to which interferes in your the neural thinking process. There you have this neurodegenerative disease, right? So these drugs has been basically acted upon these diseases, but then they have been used to treat bipolar disorders or any other kinds of neurodegenerative diseases. But it has been seen like they have also been able to work, contributing it has been able to work to those factors which are able to contribute towards tumorigenesis process. Okay. So now that we have seen various the importance of drug repurposing and with respect to cancer biology, and I have included one of the term called cancer stem cells. So we are aware of this term called drug resistance. The key player are cancer stem cells in other way around, and all of this phenomenon that has been listed here can contribute to drug resistance, or it can be in the vice versa phenomena. So at the end of the day, these cancer stem cells are we can say along with the cancer cells that are present, they basically deceive the role of this multi-targeted drug therapy or the drug repurposing. So then we go to find some drugs which can be targeted in either of this pathway, if not all, either of this pathway, which are crucial or can be the key players to trigger that particular cancer. For example, I'll give you this is a new type of cancer that has evolved. You have neuroendocrinal tumors. So they can originate from the hormonal imbalance. They will spread across from your central nervous system to your peripheral nervous system and can invade to the other by near organs. So pancreatic cancer is actually divided into two classes based on the type of its origin. It can origin from the ductal cells that are defined as exocrine adenocarcinoma. The other way is the neuroendocrinal tumors from the horn, because it's a gland and it's a digestive organ also. So it can develop the tumorigenesis from either of the source. So in such cases, we can check if epithelial mesenchymal transition or epigenetics can work in this type of cancer, and taking all those markers which are functioning in this two particular signaling pathway or two independent mechanisms, drugs, you take a combinatorial sets of drugs that can work on either of these pathways and can make it achieve simpler, that can be which might prove effective for this kind of cancer or with respect to each type of cancers. So as I was mentioning, the sets of the drugs that you can screen using tools here. If I say AI will play a major role or computational biology will play a major role in order to achieve a common age sword that can serve to combat one particular type of cancer. So you can utilize machine learning models. You can utilize your molecular docking tools. You have a number of softwares now, Schrodinger. You have Discovery Studio, BioVia Discovery Studio. You have now Autodoc Vina, which Autodoc Vina is again, it is like commonly available, but for Schrodinger or the tool that I mentioned, they are licensed. So you can utilize these tools and you can screen these drugs using AI. You can develop the mathematical model that can set your features, and you can screen down like this particular set of drugs will have reduced toxicity. It has been proven in AI platform, but then you have to validate using the in vitro and the in vivo models. So as I was mentioning previously, right, like the subclinical stage. So this is a stage where a subclinical stage of drug screening plays a major role, because this is where you are going to assess the true efficacy of this particular of those sets of drugs, which will be effective against your particular cancer type, which may not be effective, and that will save you time, resources now, as compared to the traditional or the conventional route of drug discovery. Because nowadays, since this AI tools has become so easy, you can easily design your drugs and you can reduce your timing also, right? So coming to the in vitro models, you have again two sets of culture types. One that is primary culture and the other is secondary culture. So primary culture is like you take patient cancer samples. When if you have any collaborators, you can take the help of the collaborators. You can get institutional ethical committee clearance. You can collect the tissue samples. You can culture them, and that can prove a very, I must say, a very sound scientific sound research when you're trying to check the efficacy of these drugs as compared to the secondary culture, because secondary culture is based on the cell lines that you derive from your this primary culture. You develop those cells based on your those tissue samples you derive, and they constantly divide because all is it it is basically immortal cells. So these cell lines, as you establish, you can also check the efficacy of these drugs in secondary cell lines. But I must suggest, if you have the facility of getting your tissue samples, if it is feasible technically, you can check the efficacy of the drugs taking this particular concept. As I was discussing, like AI platform is now the red queen race as compared to the conventional route. Why is it so? In healthcare, because you have n number of data sets, you can process those data, you can transform those data to your required goals, and you can utilize the statistical tools to analyze which set of data set you can generate against your particular cancer or the particular disease that you're interested. You can create a new information set, and all the while, I must say here, next generation sequencing will play a major role here, because your cancers are different based on the type of the gene mutations that you have, or the mutations which is taking place at the protein level. So the response to the drugs differs from one person to the another person, and that is why the concept precision medicine or personalized medicine therapy is evolving nowadays. And taking this help of all of these tools, you can develop newer data, newer tools, newer techniques that will be feasible also, and you can prove it technically to combat the particular disease. So considering all of these factors that we have come to know, how we should approach it here, there are some risk factors, yes, and there will be some challenges. So I have listed few of them, which are very crucial here to discuss. One is the off-target toxicity, the other is the selection bias, and the other is the intellectual property barriers. So what do you define by off-target toxicity? So as I was telling, like you have seen like multi-target therapy, right? The basic notion of drug repurpose is you have this set of the drugs and you are trying to achieve those drugs should be able to work against that particular disease type. Now that it will have several targets to work with, and that's how it will be effective. It might induce some other side effects that has not come into the picture till now. Using this concept can also come up. So that is one of the factor here. And the other way is selection bias. Yes, as I was mentioning you, like the drugs response will vary from one population to the other. So the personalized medicine approach is coming here, but then it may not be applicable to the other group of people. Okay, so while you're selecting those sets of drugs, we have to be careful. We can consider that those drugs taking to the genetic profile. For example, as we saw, like a few drugs are getting metabolically active in terms of SNPs. So you can consider the SNPs which are responsible for showing its toxicity or which are responsible for its activation state. So based on this complete profiling, you can take the new sets of the drugs. And the other is intellectual property barriers, because you remember while I was speaking about the pros of the drug repurposing over the conventional drugs, it is based on those failed or the banned drugs. So companies which have produced this much efforts to produce those drugs and they have gone to the market and it might have undergone retraction. So they won't try to reveal the mechanism behind it. Why was it not effective? Why it has failed? So again, it comes the conflict of interest. So that restricts the usage of these drugs to the further level. So, in line with this particular concept, we are all aware of this terminologies: CRISPR-Cas9, silenced RNA, miRNA based therapeutics, precision medicine, as I mentioned, or pharmacogenomics. And the king is also here, like the single cell based therapeutics. But then, considering the complexity of the tumorigenesis concept, what is it and who should lead is still questionable. So that's all I wanted to discuss about in today's JC. And thank you one and all for listening so patiently to my talk. And these are some of the references that I have consulted. And I am happy to answer your questions now. I'll be happy to answer any questions, any basic questions that you have also. Any fundamental questions, even if you have, you're happy to ask it also.
Hello.
Hi, ma'am. Hi, ma'am.
Yeah.
Hi, ma'am. Thank you.
Hi.
Hi, ma'am.
Very nice presentation. Yeah.
Thank
Just
you.
a
Thank you,
few
ma'am. Thank you
things,
so much.
just very basic thing that I wanted to ask. Like you said that
please,
AI
ma'am.
is definitely helpful in this, but
Yes,
how
yes.
well do you think the predictions that we do through AI are applicable to the practical bench based, मतलब कि जो हम experiments
yes.
जो करते हैं,
करते हैं.
and as compared to that, the simulation studies that we do, that how
Yes,
do you
yes.
think that the proportion, or just a
Proportion.
common view of yours, like
Yes,
how do you think
yes,
it's
ma'am. Yes,
applicable?
ma'am. Yeah, I understand. I agree with you completely. What I wanted to mean was like AI tools and techniques have reduced the time and the resources that are being implemented, right? Because when we do in wet lab, we begin from a scratch. Huge amount of resources are being exploited in that case. But then, how does AI help is, although it is just a prediction model, it gives us an at least a preliminary idea, like what all should be screened or what all should be utilized. And taking those information, that's why I have mentioned, like the fundamental efficacy of those drugs or those compounds will lie only when we come to the subclinical stage, ma'am. It is not like I'm telling purely AI should revolutionize this area, because there will be some flaws always behind the computational prediction, the features that we are giving, or the data sets that we are putting into it. So there will be some flaws there. As of my opinion, I cannot tell that it should be completely reliable. So it is
Yes,
only
but it is
yeah,
just
yes,
that,
please,
ki, like,
please.
that in a cohort, that when we see in a general small population also, we get to see a loads of variation.
Yes, that's why I mentioned,
Yeah,
ma'am.
so
While we screening, also we should be very careful the data sets that we are providing while running the AI model, and
okay.
considering, yeah, ma'am, and considering the fact, for example, I'll give you like an instance. So There is one paper that I went through. When you have hundreds or 200s of drugs that you want to screen with all the features all the features, like especially with respect to safety, then the around to 20 or 25, which is doable which I must say, like it is doable on the on the in vitro and the in vivo assays, as compared if you take a bulk of compounds, right? So that's what I mentioned, like it is not completely reliable, but then it is one way helping us to give some preliminary ideas which we didn't have into the picture before, when AI has come into the market, ma'am.
Okay, nice. Thank you. That was very nice. A nice works
Thank you.
and study done. Hopefully, get some better
Yeah.
results with studies.
Yes. Actually, the thing that I have presented here, it is related to my PhD work. I could not show my PhD work, but whatever I have presented here, it is related to my area of interest that I have pertained during last few years. Yeah. Any more
Okay.
questions? Any
Okay, great.
simple questions?
Thank you.
Yeah.
Thank you,
Yes.
sir.
Thank you, ma'am. Thank you, ma'am. Thank you so much. Any basic questions, I'll be happy to answer.
Hello.
Hello.
Okay, thank you, Adriana.
Hello,
That
Agu.
was a very good. Thank you. That was a very wonderful presentation. Drug
Thank you.
reproposing is about the center of my research interest, especially nutraceutical, which is also part of drugs reproposing, and
Yes.
it's gaining weight in discussion around the globe. So I think one of the challenges with young scholars in this drug reproposing is when they choose to take bioinformatics approach, find out that sometimes the softwares are not freeware. So, and sometimes the ones that are
Yes.
freeware, they lack the knowledge to install them. And some are more flexible using Ubuntu or Linux, whereas like in my own end, we use more of Windows. So if there would be ways to maybe in the upcoming to look at the bioinformatics softwares that can be good in using in silico identification of these the characteristics of these chemicals that can be repurposed in drug design, just through the use of
Yes.
the tools and install them, their approach
Yeah.
in drug proposing, that will make a whole lot of sense. Thank you very much. Thank you. It's a
Yeah.
wonderful.
Thank you for adding that point. Thank you for adding that point. Yes, I completely agree with you, Agu. Thank you so much. Anyone else? You can ask your question, I think, if you have. Kavya.
Yeah,
Hereafter,
am I audible?
yes, Anuj. Yes, please. Yes, you
Yeah,
are
I have
audible.
a
Yeah.
similar. Yeah, I have a similar question or query like the Agu that because as a newcomer or a beginner to this kind of computational analysis and all that bioinformatics, so how do we get informed about that? What are the softwares that are freely available or like that resources? Is there any particular website or portal that we can refer to get an idea about?
Yes, I mean you can go through it. I you see once if you I'll tell you firstly the first freely available software is Autodoc Vina or Autodoc itself. It's freely available software, and my part of the study. I'm sorry that I could not share the data with you all. So I did it in Autodoc only. So it provides a very simple protocol to it provides a very simple protocol to perform this the docking analysis. If you want feature, I can share the protocol also. And yes, I mean in nowadays if you go through in online, you can just simply type the freely available softwares for docking, but the most commonly widely used is Autodoc, I must say, and that is widely accepted from publication point of view also. So you can very well go with that. But for simulation, you have to run it in Chromax or Desmond, some licensed software. Because, and one more thing is, simulation, you should have some supercomputing facility, because it takes huge amount of time to run. And what if your protein and ligand complex is also big enough? So in that case, it restricts. So you should look out for people who are working in a similar field, and that will help you to reach target your goal. I hope was it helpful, Anuj?
Yeah, thank you. And it's a great learning experience to listen to this kind of talks. Thank you so much.
Thank you so much, Anuj, for joining. Yeah, any more questions? I'll be happy to answer. Manjari ma'am?
Yeah, I think that's it. Maybe we can end the call. Thank you all.
Yeah, maybe we can have a photo, ma'am, if it's fine for you.
Sure.
Yeah, Razul, are you there? Yeah. Yeah, I request all the participants, you can just switch on your camera and we can have a photo for our first journal club session. And I would also like to add, we will have monthly twice this journal club session, and it can be from UG, PG or PhD students, whoever is interested. Please put your topic whatever you want to present 1 week priorly with the meeting link. Yeah, we can join for a photo. I just request everyone to switch on their cameras. Pradeep, Anuj, Dheeraj, Malvika. No one is. Yeah, I request everyone to take a photo. Okay, Dheeraj says he can join. He can switch on. Malvika, Agu Peter, Meherin.
Yeah, I think everything's giving just quality.
Yeah, it's okay then, ma'am. Fine. Okay, then we will call it for the day. Yeah. Thank you everyone for joining the session and making it lively and interactive. Thank you one and all.